So we're on chapter 12, section 2 of your biology book, and we're talking about bacteria, and we're going to go on to talk about viruses. So here on table 12.5, this is a list of, as we go through this list here, of diseases that are caused by bacteria. Bacteria, anthrax, botulism, cholera, diphtheria, food poisoning. So you have these food poisoning caused by staph infections. There are E. coli infections, gangrene, clostridium, gonorrhea, leprosy, Mycobacterium leprosy, Lyme's disease, necrotic fasciitis, rheumatic fever, scarlet fever, strep throat, toxic shock are all streptococcus. So we have staph and strep. See all the different kinds here? We go down peptic ulcers, Helicobacter pylori, anyway, pink eye, conjunctivitis, the plague, both bubonic and pneumonic forms of the plague. Rocky Mountain spotted fever, rickettsia, syphilis, tetanus, tooth decay, dental caries are streptomutans, tuberculosis, Mycobacterium tuberculosis, typhoid fever, salmonella typhoid fever, and typhus. They're not the same. Rickettsia. These are two different things caused by two different bacteria. So there's a lot of bacteria, but now there's something. Viruses are the most common of all, even more common than bacteria. So let's talk about viruses. So viruses they are like the flu bug, intestinal bug, cold, and as you see, they're very difficult to classify as a living thing at all. And they're very difficult, as for a cold, to control. You usually get them. They're hard to develop any cure for some of them. Viruses are more of just a molecule. Let's go over here to look at some of these viruses here. Look how strange these guys look. So let me move down here a little bit. Structure of viruses. So they're a molecule of DNA or RNA, actually. They're just one single. They're very tiny. They're enclosed in a geometric protein called a capsid, like a capsule, but it's called a capsid. And some capsids are surrounded by lipids and proteins and carbohydrates, but they're much smaller than bacteria. So they go through replication, make new viruses, of course, and they're manufactured and assembled by infected cells. So they're basically going to attack the cells, and most of them go into the cells. The ribosomes, let's look at these pictures here. These are different ones. Here we got the capsid here. We have this is the herpes virus here with the envelope there and the genetic material in here. Here's the adenovirus, is a simple virus here. You see they have these spike proteins, kind of like COVID here. And they have genetic material or DNA or RNA, identification tags. The spike proteins are its identification tags, and this geometric capsule here. And then we have down here at the bottom, we have this helical virus, tobacco mosaic virus, genetic materials up here, and the helical protein capsids, like I say capsule, but it's capsids up there. And then we have this weird looking spaceship thing. This is a complex virus, and the genetic material on the top and the protein capsid. Then you have the DNA injection mechanism there, and this is a bacteriophage. T4 infects bacteria. So, wow. So very strange. As for the replication, they make new viruses. They have these, as for the ribosomes, the factories are programmed by RNA, for the RNA blueprints, and their new protein, the RNA produced by the nucleus, and they ship them to the ribosomes, and the ribosomes start producing protein to make new viruses. A virus has to have a host, though. A A typical virus is essentially a capsule of genetic material for a new virus. Virus invades. As when a virus invades, the genetic material takes control of the cell. So we're talking about how it invades. It goes to a cell. The genetic material takes control of the cell, like getting in the cell. And the cell makes copies of the viruses. The virus's genetic material takes over in the, a lot of times, RNA of the cell and assembles this new virus that's ejected from the cell. Sometimes host cells, all of a sudden, they will just burst open and the virus will go all over the place. Viruses cannot reproduce independently. They have to have a host, and they're merely just the set of instructions that are encoded DNA or RNA, and they copy, in the cell and spread in those living cells. Diseases, like the virus does not carry on the metabolism or life activity with the disease, so it's actually just breaking the cells down. Virologists study viruses, and they're very uncertain if these things are really living things. There's things like viruses that cause colds, sores, warts, rabies, smallpox, chickenpox, yellow fever, polio, mumps, measles, influenza, mononucleosis, and colds. Some contribute to cancer. COVID-19, of course, and Ebola, and West Nile, and AIDS, and chickenpox. So a lot of virus. The HIV/AIDS virus is a retrovirus, which gets into the DNA. So it takes the DNA blue point into this host cell and causes AIDS. Wow, it's a lot of different viruses. So let's look here how it's doing it, the life cycle of a virus here. So we have the virus enters the host cell. So here's this little virus, it's coming on in, and then the virus is free in the cytoplasm of the cell. And number three, it goes for number three here, the virus here is free in here. The capsid opens, the capsule, like, of a spaceship or whatever, but it's the capsid opens, releasing genetic material of the virus, the DNA or RNA come out. And there's the little DNA or the RNA come out. And these genetic material, this, it looks, DNA is like coiled, and that's RNA is like a half a coil, comes out. And the viral genetic material programs cell's own machinery to manufacture. So it makes the cell manufacture the virus parts. It interrupts the cell. And then the new virus assembled from these components. So they find the cell's own machinery, and they take these come in here, and then they make their new viruses here. And then over here we see, number 6, they assemble viruses, assembled viruses exported from the cell by budding. So they come out of the cell here, and then new viruses are free to infect other cells. Wow, quite a deal. So you see how a virus reproduces. I've studied AIDS, and the HIV virus is the virus for AIDS, and it acts a little bit different because it gets into the DNA. It binds to the receptor, like you'll see a virus do this. It binds to a receptor to T cells. So it affects our T cells, and T cells are very important cells in our body. They're leukocytes, they're our white blood cells, and if we don't have enough T cells, we're going to have a hard time fighting any infections. So someone that has an HIV virus attacking the T cells, they'll get it when they come down with AIDS, which is the disease. AIDS is the disease, autoimmune deficiency syndrome. They will get all kinds of sicknesses that most people don't get, like their white blood cells are not fighting off certain sickness. They'll get cancers that are very rare. They'll get thrush or funguses in their throat that are very rare. All kinds of strange things because they can't fight off these diseases. So basically how the HIV gets in, goes through the same thing as it's going here, but what it does, it has a reverse transcriptase, an enzyme, and so it goes to the RNA of the cell, and in the RNA, the messenger RNA of the cell, it gets into the genetic material, into the DNA of the host. And so going into the host's DNA, it starts this genome integration, what's called, where it synthesizes DNA and travels to the cell's control center and screws up the whole cell. And basically it goes through replication and everything else with the cell and making cells there. Finally, the same process happens with all these little viruses that have gone into the nucleus of the DNA, and they're called retroviruses. And then they release and push out. So it's a terrible, disease. So, but because it's so different, because it goes into the DNA and it replicates that a different way with this reverse transfer, they call it a retrovirus. So let's go on to the next here. So that's about the viruses, but let's go and finish this chapter. Let's go down here to the selected viruses here. So here we go with our virus list. So first we have the retrovirus HIV, and it uses the RNA, messenger RNA, and gets into the DNA. Chickenpox, the virus genetic material is DNA, so the DNA of their own, and it's a herpes virus. Then we have cold sores, DNA of the a virus. Common cold, different rhinovirus is RNA. COVID-19 is RNA, it has the RNA. Ebola, RNA. Influenza goes the RNA. Measles, RNA. Mononucleosis is the herpes virus. Epstein-Barr virus is DNA. Polio, RNA. Rabies, RNA. And warts, papillomavirus, DNA. West Nile, RNA. You can see, and just this, that's when they say the genetic material, that's the genetic material in the virus. But it can infect the parts, like I said, AIDS affects the RNA. It's like a messenger RNA that's disguised and gets into the DNA of the cell. So now let's go here. We're going to talk about, I'm going to move over here so you can see what here we're going to talk about the mycotic diseases. The fungus, which is athlete's foot, ringworm, comes from a fungus, and it gets your skin conditions of red, flamey, flaky skin. Also, thrush of the mouth in infants. This is an infection of this fungus in your throat. Dandruff can also be a fungus of the scalp. Yeast is a type of fungus, and it can get into your scalp and irritate it. And you have to have an antifungal shampoo if that happens, right? So, and going on to the helminthic diseases, which are worms. Worms again, roundworms, tapeworms, fluke. Trichinosis is a type of hookworm that goes in there. And then we're going basically going in thinking about protozoa, these single cell protozoa. Malaria is the protozoa that infects 150 million new cases each year around the world. That's a lot of malaria. And so it's a protozoa. Amoebic dysentery and African sleeping sickness, and a few others are protozoa, single cell, these single cell cells that go in here. So let's go on here to the next part, which says the spread of infectious diseases. So all right, here, see if we're going to talk about this guy here in just a little bit. So I'm putting this picture, but before we talk about him, let's talk about the spread of infectious diseases. Air, some spread by air, by coughing and stuff and getting the air droplets. Some by food, some are in our food, some in our water. Sometimes as a contact with that diseased person, person-to-person contact, or a contacted disease, or a contact disease person who is not even ill, somebody that's not even sick can be a carrier and give us the disease. Also, insect bites and animal bites can spread disease. Most transmission are by human transmission or by animal sources. So, and of course we have, again, coughs and sneezes are droplet infections, and also singing and talking can spread by droplets too. Direct contact on your hands, and how hand washing is very important. And others, okay, could be some carriers of disease here. So let's talk about this lady up here. Let me see if I can find what they wrote here on this. Don't see it right here, so let's go back. Where we got droplet infections, we got carriers. Okay, this is the story on the carrier right here. Her name was Typhoid Mary Mallon, lived in 1869 to 1938. What was about Typhoid Mary? She didn't have any symptoms and she had typhoid. She lived, she was immune. She was immune to it. She wasn't sick, but she could infect others. So she moved around, Typhoid Mary, from household as a cook. She was people's cook. So she'd handle the food. And so the people in the houses would get sick. And she even went into the New York maternity hospital, and tons of people got sick in there. And 3 people died and scores of others got sick. And so finally she was arrested and she was quarantined for 25 years because she was a carrier of typhoid. That's an amazing story. So you can be carriers of certain diseases, especially typhoid. There's also indirect transmission by insects. Malaria is transferred by what? Mosquitoes, right? Mosquitoes. Malaria is mosquitoes. So a mosquito is a vector. So whenever you have something, an animal or an insect, they're called a vector. So a mosquito would be malaria. Also, yellow fever is another type for mosquito. And a vector, like I said, that transmit could be a mosquito. That can be a louse, like lice. It could be fleas. A flea vector was with the bubonic plague. Fleas were actually on rats. These rats had these fleas, and the fleas carried the disease of the bubonic plague and killed many people, but this bacteria was on the flea. And there was a lice vector for typhus at one time, with a lice vector on lice. So subcutaneous contaminated, like food poisoning, salmonella, you can actually get that, from not wiping off the table very well. Like say you're making meat, some kind of, fish or something, and then you don't wipe off, and you basically, had a cutting board, maybe cut vegetables on it too and eat the vegetables. You can get food poisoning, salmonella. So, and also from contaminated water and contaminated blood. Contaminated blood is a big thing too. So, and then of course down here we have sexually transmitted disease, and there's lots of sexual transmitted disease. Venereal diseases are called when having sexual relationships. In 2018, 26 million new cases of STDs. Half of those were young people between 15 and 25. So, promiscuous and homosexual, these all STDs are up, of course, and AIDS being one of them. And the incubation period before it starts. So sometimes people have carrying the disease and they have an incubation period and they're not sick yet, but they can transmit certain diseases. So prevention is very important, of course. That's basically, if you're sick, if you're sick, we don't want to go to school or go be around people, and just and big prevention is hand washing. It talks about an epidemic. is when the disease breaks out rapidly with a lot of people. And then there's pandemic. Pandemic is like a large portion of the whole world. As they said, basically, the COVID-19 was a pandemic all over the world. So there's other pandemics like the flu in 1918 was a pandemic. The plague in the Middle Ages, the bubonic plague was a pandemic. And of course, AIDS was a pandemic too. So let's go on here. Okay, now let's go back to this guy because I love this story here. Ignaz Semmelweis. That's how you say it. Ignaz Semmelweis. So Ignaz Semmelweis was basically, he was one of the first ones to really, let's see if we can grab this here, find out about hand washing. You know, before that, nobody washed their hands, and even the doctors didn't. So Ignaz Semmelweis lived in 1818 to 1865. He was a Hungarian doctor, and he was the first to think hand washing is important in the hospital. So he worked in this Vienna hospital where there was a lot of maternity patients there having babies these ladies were healthy. But 25 to 30% of these patients that were having babies died. And they died of types of infections. And then the women that were having their children at home did not have a high rate. They were living. So why were all these women dying, he thought. So he basically realized that the women were being examined by medical students. But before the medical students got there, they went and did autopsies on dead bodies. And they did these autopsies and they didn't wash their hands. And then they went and examined these women, and these women were getting sick. So you can imagine. So they didn't require people to wash their hands back there. And so he decided, I'm going to make everybody wash their hands. And he found this antiseptic type soap that he had. At that time, they didn't know it was antiseptic. We found something in washing their hands. And the mortality dropped from 30% to only 1%. So he was so excited. He went around and told everyone how they needed to wash their hands. They had to wash their hands. And he said, if you don't wash your hands, this is important. There's something on your hands. And that was before they even knew about germs and everything on their hands. So everybody got mad. You think they all said, oh, that's good, we should wash our hands. Nope. They relieved him of his position. They were mad at him. And then they just all went back to doing the same old thing. And of course, it went right back up to 30%. Those women were dying again. Though he was right, he met harsh hostility and ridicule by the traditional medicine. And really, he didn't know hand washing was so important. They didn't know until after his death. And he's the one, he said he knew it had to do, that he could make the disease go down, the mortality rate go down to way less just by these guys washing their hands. So pretty amazing. Pretty amazing. So let's go back to where we were before, and I was talking about prevention. Well, prevention being hand washing and cleanliness. And hand washing and cleanliness in the Bible was way before. In Leviticus, they basically said the priests had to, if there's any infection, they had to isolate, and they also had to wash. They had to wash their hands and had to wash their bodies sometimes. So when the bubonic plague struck, guess who didn't die? The Jewish people, because they had hand washing and they believed in the Bible and hand washing. So they were persecuted, in fact, because of that. So, because until late 1800s, doctors and people did not wash their hands. So all kinds of diseases were spread by that. It was hospitals were breeding grounds for that. And so another thing to do is to avoid unnecessary contact with people that are sick and to stay home. In Leviticus 13:46, it said, if you're sick, you need to quarantine yourself for 7 days, and they did in the Bible. Disinfectant contaminated objects and chemicals. So we disinfect things with chemicals, right? Or radiation. So chlorination of drinking water is important to prevent cholera and typhoid fever. Cooking food thoroughly, so prevents food poisoning. Cook away those bacteria, if there's any bacteria. Sterile surgical instruments, all important. Disinfecting is important. And then avoiding or eliminating vectors. That means, what does that mean? Malaria. So when they were working on the canal, Panama Canal, they had to figure out what was causing malaria, because all the workers that went there, they would get malaria. They found it was the mosquitoes. So they went in, they thought if we could kill the mosquitoes, so they went out and put forth mosquito, basically insecticides and everything to kill all these mosquitoes, and that stopped malaria. So to kill the vector, get rid of the vectors that are transmitting the bacteria. So, and now we have, of course, vaccinations. Vaccination is a substance that stimulates the body to develop acquired immunity. And so the body will produce antibodies, either months or years. Also, there's herd immunity, when everyone goes through and everybody gets sick, and then everybody gets immune. But going back to the vaccinations now, if we think of COVID vaccination, it's not really a vaccination. It's actually a genetic. It's actually a genetic, how they go in with the RNA, messenger RNA. So a vaccination is a substance that stimulates the body to develop acquired immunity, basically through antibodies, and that is not the same thing. Just a note there. Joseph Lister. Let's go to Joseph Lister, and then we'll be finished with this chapter, or this section, I should say. Here's another guy, Joseph. So, here's some diseases that were fought with vaccines. Chickenpox vaccine, cholera. Chickenpox, if you get the vaccine for life. Cholera, 6 months. Diphtheria, 5 to 10 years immunity. Hepatitis A, hepatitis B, 20 years to life. Influenza, 2 to 12 months. That's flu. Measles, if you get the vaccines for life. Meningitis, mumps for life. Pneumonia, pneumococcal, polio for life. Rabies, rotavirus, rubella is a type of 3-day type measles, kind of called rubella. Tetanus, 5 to 10 years. Tuberculosis, typhoid fever, 2 to 5 years. Whooping cough, and yellow fever. So let's go to Joseph. Joseph Lister. So he lived in 1827 to 1912. He was a British doctor, and he was one of the first to use antiseptics, chemicals that kill bacteria. His father actually worked for the Royal Society, and he was a scientist too. He put forth a new kind of microscope, So Joseph was raised as in the area of science. And for 9 years, he did a lot of studying, but in 1861, he did experiments with antiseptics. And so basically, he was the first to find that through the air that it was carried, that something was carried on the surgeon's hand, and that first, basically, he had an experiment with antiseptics. He had this, he was experimenting with bacteria. He thought, does pollen dust cause infections? In 1865, he was thinking about microorganisms that would cause disease. And so then he thought, well, maybe through the air they're carried, or maybe they're carried on the surgeon's hand. So he used carbolic acid, phenol, as an antiseptic for surgery, and the mortality rate from people dying went from 45% clear down to 15%. So, but it took many years for others to really accept this. He was he was a Christian man, very unassuming man, but he was the first to talk about antiseptics. So that is it for this chapter. So wow, there's a lot of things that we need to trust the Lord, and the Lord can heal too. So we're going to go on to protection against disease, and we'll be finished with this chapter.